Today, we will do the review of the chapter we have already done, which is biotechnology and its applications. So, when we were in biotechnology and its applications, what we did first, that was we did the difference between the hybrid and the GMOs, and then we did what I want to repeat with you is the use of biotechnology in agriculture. Biotechnology in agriculture. What is the use of biotechnology in agriculture? In this, the first thing we did was BT, Bacillus, use of Bacillus thuringiensis. This is a bacteria. How this bacteria was used in the field of agriculture to develop the GMOs, the GMCs, to develop genetically modified crops like BT cotton, BT brinjal, BT corn, etc. Now, what is the specific property? Why this Bacillus thuringiensis was used in the field of agriculture? This Bacillus thuringiensis, it has a gene in it, and that gene which is present, I'll use the short form BT for Bacillus thuringiensis now. BT has a gene in it. It has a gene, and that gene is known as cry gene. We call it cry gene because it produces the crystal protein, which is cry protein. This cry gene, it undergoes transcription. The two processes it undergoes: transcription and translation, and it produces a protein which is cry protein. Specifically, this cry protein is thuricide. Thuricide. Thuricide is a substance which is produced by this cry protein, and this is a toxin. This is a toxic substance. We also call it as endotoxin. This is an endotoxin, the toxin which is produced within the body, endotoxin. This endotoxin is toxic to, this is toxic to the caterpillars. It is toxic to, basically we can say, pests. It is toxic to the pests. Now, specifically if we talk about which pests, like caterpillars, mosquitoes, flies. Okay, so this is and many more insects which harm the crops, and many more insects which harm the crop. This way it is toxic to it, and that means these pests are killed. When we say that it is toxic to these pests, that means these pests are killed. So that means the crop is saved from the destruction caused by these crops. So the crop is saved this way. Now, this particular property of Bacillus thuringiensis was used by the researchers who were dealing with biotechnology. And what did they do? What did they do during this process? These researchers, they took out the cry gene. This cry gene was taken out from Bacillus thuringiensis, and it was introduced in the plants. It was introduced in plants. That means now what we have got? We have got transgenic plants. We have got These plants in which this cry gene is introduced, these are transgenic. When these plants, which are now transgenic plants in which the gene is introduced, already the gene is there. Which gene is there? The gene which produces endotoxin thuricide. That gene is there in the plant. Now these are the transgenic plants. Now this gene in these transgenic plants, this undergoes the process of transcription and translation and produces cry protein. Here the protein is thuricide, which is toxic to the insects. Thuricide is produced. Now that means in these plants, which are now transgenic plants or BT plants, also known as BT. Suppose we take here cotton plant, so it will be BT cotton plant. So in them the gene has been introduced, and in them this gene, in these plants, this gene undergoes the process of transcription and forms the cry protein. This, what is this cry protein? This cry protein is this thuricide. That means the toxic substance is already there in the plant. Now this is in the inactive form. When the pests, they attack the plant, when the pests attack the plant or attack the plants, what happens? They will feed on the leaves, they will feed on the other products of the plants. So what happens? This thuricide enters their body. Thuricide, which is the cry protein, enters the body of pests. And this thuricide is toxic to the pests. That means this thuricide is not suitable for the pests. It kills the pests. Now what happens? How does it kill the pest? These pests, the medium in the gut of the pest, gut or alimentary canal. The alimentary canal or gut of these pests, when it reaches the alimentary, when this thuricide, when it reaches the alimentary canal or gut of the pest, there, if the gut is like this, their gut is like this, their alimentary canal or gut is like this, it has epithelial cells. These are epithelial cells which are present on their gut. The lining of their gut is made up of epithelial cells. So this thuricide, how it harms this, first of all, in the gut, it becomes activated. When this thuricide reaches the gut, it becomes activated because the gut has an alkaline medium, and in the alkaline medium, it gets activated. Now this activated thuricide, now see, in the alkaline medium, what do we get? We get activated thuricide. We get activated thuricide. Now, what this activated thuricide does? This activated thuricide makes pores in the epithelial cells of the gut. See, the pores has been made. So you can see pores are made in the epithelial lining, epithelial cells of the gut lining. The lining of the gut has epithelial cells, and the epithelial cells, on the epithelial cells, the pores are formed. When these pores are formed on the epithelial cells, then what happens? Water enters these pores. Water is there in the body. The water enters their pores. When the water enters these pores, the epithelial cell swells. The epithelial cell swells. When this water enters the pores, the epithelial cell swells, So swells, and when this swelling reaches the maximum limit, they burst. And due to the bursting of epithelial cells, bursting of epithelial cells kills the pests. Bursting of epithelial cells kills the pests. And this way, the pests are killed and the plants are saved from the pests. So we got, so this is how the people who were working with the biotechnology, they were able to obtain pest resistant plants. Pest resistant plants, these are also known as transgenic plants or genetically modified crops. And the names I have already told you, like the BT has been BT gene has been introduced in case of corn, in case of cotton, in case of brinjal, etc. Now, amongst all, BT cotton was the first transgenic crop to be introduced. BT cotton. This was the first transgenic crop to be introduced, and this was the first which got approved by the committee, which was approved by Government of India. Government of India also approved this crop, and this was the first crop which was approved by the Government of India, and then now this is being used extensively in the country. So this is the BT cotton, which is first of all, which was first of all obtained by the people who were working with biotechnology. Then it was approved. So this was about the BT, the use of Bacillus thuringiensis in developing pest-resistant plants. Now we come to another use of biotechnology in the field of agriculture, where I'll tell you something about the flavor saver tomatoes. Flavor saver tomatoes. Now, what are these flavor saver tomatoes? You know the name flavor. The spellings are given like this when we say flavor saver tomatoes. Flavor saver tomatoes. You know, usually what happens, not before, I mean, not now. Nowadays, the biotechnology is in use. I'm talking of the time when biotechnology was not in use, and the tomatoes which was obtained from the before the use of biotechnology. Biotechnology in the field of agriculture, the post harvest, the shelf life of post, of course, shelf life is only after post harvest, so it's not necessary to write post harvest. The shelf life of tomato was very short. Why this was? What do we mean by short tomato life? A short shelf life, that means after plucking from the field, say within 2 or 3 days, they used to get spoiled. Now, what is responsible for spoiling or decaying of tomato? Now, tomato has, the wall of tomato has a substance in it. Wall of tomato has a substance in it which is known as pectin. This is a hard substance and it gives firmness to tomato. So this is hard substance and gives firmness to the tomato wall and keeps it fresh. That means it is responsible for the freshness of the crop. It, it is responsible for the freshness of the crop. Now what happens? Tomato, tomato itself has a gene which is known as PG gene in it. This gene, after translation, after transcription and translation, produces a substance which is known as polygalacturonase. This polygalacturonase has a property. What it does, it dissolves pectin. This polygalacturonase dissolves pectin. That means if pectin is dissolved, that means the firmness is lost. If this pectin is dissolved, dissolution of pectin is responsible for spoilage of tomato. So if this pectin is not there, then the shelf life can be increased, because what will happen? The pectin will not dissolve and the shelf life of tomato can be increased many folds. Okay. Now what was done to make, and such tomato can be known as flavor saver tomato. Now how this was done? How this flavor saver tomato was produced? There was a biotechnologist, and the name of biotechnologist was Mack. Mack was the biotechnologist, and this gentleman Mack, what he did, the name of the biotechnologist was Mack. What he did, he took out this gene which is known as PG gene. It was taken out. PG gene it was removed from the plant. Removed from plant. It was removed from plant and was taken to the lab. Taken to the lab. In the lab, it was taken to the lab. In the lab, this PG gene removed from the plant, taken to the lab. Now, what was done in the lab? This PG gene was allowed to undergo transcription. So, transcription. As a result of transcription, what is produced? RNA is produced, messenger RNA. So till here it was done in the lab. PG gene removed from the plant, taken to the lab. In the lab, PG gene was allowed to undergo transcription when RNA was produced. Now this RNA was, allowed to undergo a reverse transcription. It was allowed to undergo reverse transcription. As a result, what was produced was complementary DNA. This DNA was complementary to this RNA. That means their nitrogenous bases were matching somewhere. Their strands were matching. Okay, so this was complementary to this. Now, what is reverse transcription? The central dogma is DNA. From DNA by transcription, RNA is produced, and from RNA by translation, protein is produced. Now, when we say reverse transcription, till here, that means RNA was produced, but from RNA, protein was not allowed to be formed by the process of transcription, rather by the process of translation. Rather, this RNA was allowed to undergo reverse transcription, and as a result, complementary DNA was produced. So this complementary DNA, this DNA was complementary to this. Now what happened? This complementary DNA which was produced, it was introduced in the plant into its genetic constitution. It was introduced in the plants. So now the tomato plant which was formed had cDNA in it. That means what this gentleman Mac did, he introduced this cDNA in the plant. So now tomato plants had complementary DNA in them. This gene, cDNA gene in them. Complementary DNA in them. Now, what happened when in these plants where complementary DNA was present, the normal gene was also present. Normal PG gene was also present. This is present naturally in them. This normal PG gene is present naturally in them. That means now the tomato plant has this gene which was naturally present in them, plus this complementary DNA which was introduced in them by the researcher Mac. So such plants, such GMOs or such transgenic plants, in them, see, naturally this P gene, it will undergo transcription. This is the naturally what is happening. It undergoes transcription, and as a result of transcription, RNA is produced. Now, when this RNA is produced, as I told you, there is a complementary DNA which is complementary to this RNA. So when this RNA is produced, it will not undergo translation. That means the further process, which is translation and the formation of polygalacturonase. Normally, this is the process. Normally, this is the process which is going to happen. But this doesn't happen, and at this stage, the process is stopped because once this RNA is formed as a result of transcription of PG gene, this doesn't go further. Rather, this complementary DNA combines with it, and the further process is blocked. Further process is inhibited, and because of this inhibition, this polygalacturonase is not formed. And when polygalacturonase is not formed, that means no dissolution of pectin. And when pectin doesn't dissolve quickly, so what will happen? When the pectin doesn't dissolve, the tomato remains firm. That means shelf life of tomato increases. This is how the shelf life of tomato increased by making flavor savor tomatoes. Okay, is it clear to you all, children? Yes, beta. Is it clear to you all? Yes, bache. I want your response. Okay. Now another one. I'll tell you something about the Millardogyne incognitia, RNA interference, how a gene is silenced. Now, the next process which I'm going to describe is RNAi. Here I means interference. How this RNA is interfered, how this RNA is not, does not proceeds further in its process. So how this is done? There is a nematode, and the name of the nematode is Miladogyne incognitia. So there is a nematode. The name of the nematode is Miladogyne incognitia. And this Miladogyne incognitia infects roots of tobacco plants. And as a result, when the roots of tobacco plant is infected, there is less production or less or no production. So productivity is affected. Less or no production. This happens. Now what to do so that the gene, so that this nematode does not infect the plant? For this RNA interference was done. Now what happens in this case? This nematode Miladogyne incognitia, this has a gene in it. Normally, this has a gene in its body, and this gene undergoes the process of transcription and translation. And as a result of the process of transcription and translation, it produces a protein. And this protein is required for the growth of nematode. Required for growth of nematode. That means if this protein will not be there, this nematode will not grow. This nematode grows only if the protein is, the gene is there, and that gene is producing protein, and that protein is required for the growth of nematode. So if protein is not there, the nematode will die. So this concept was first of all studied by the biotechnologist and was used. So normal growth is only in the presence of this. Now there is a concept, a basic concept, that all the eukaryotic cells, there is a basic concept that when any gene is introduced in any eukaryotic cell, when any gene is introduced, they produce a defensive protein, and that defensive protein is known as dsRNA. That means double stranded RNA. This protein is produced, the double stranded RNA. Now, what was done by the scientists? That what they did, how did they protect the tobacco plant? This is the basic concept which you have to keep in mind, that whenever a gene is introduced, the eukaryotic cell produces double-stranded RNA, which is a defensive protein. This basic concept you have to keep in mind. Now what happened? What the scientists, the biotechnologists did, they took out this gene which was producing the protein for normal growth of the nematode. Nematode was taken, and from the nematode, the gene, which was, this gene was, which gene? Which was producing protein for normal growth, for its normal growth. This gene was taken out and introduced in tobacco plant. This gene was taken out and introduced in tobacco plant. So now this tobacco plant was a transgenic plant because in this tobacco plant, the gene from nematode, from Melodogyne incognitia was introduced. Now what happened? As this gene was introduced in the tobacco plant produced dsRNA. So now the tobacco plant in defense, when this gene was introduced, tobacco plant is a eukaryotic cell, so it produced dsRNA. Now that means the tobacco plant has in it the dsRNA. When this plant was infected by nematode, when this plant was infected by nematode, which nematode we are talking about? Melodogyne incognitia. This nematode, when it infected this plant, so what happened? Nematode infects this plant. So this nematode, it enters the plant, and in the plant, what it will do, it has that gene, and that the gene which produces protein for its normal growth, it has that gene. So this gene will undergo translation, transcription, and translation, and will produce first it will undergo translation and will produce RNA. This RNA produced, this RNA produced, this RNA will not undergo. Normally, what will happen? This will undergo translation and will produce protein, and this protein will help in the growth of this nematode. But this will not happen. Here, the process will be blocked because dsRNA is also already there, and this RNA will combine with this dsRNA. It will combine with this RNA, and further process is blocked. That means this is silenced. The further the gene is silent, the gene which was there in nematode, it is silenced. It is not allowed to undergo the complete process of protein formation. This protein which is required for the growth of Melodogyne incognitia is not formed because it is blocked here. Why it is blocked here? Because as a result, the transcription RNA which is produced, it combines with the dsRNA which is already there in the tobacco plant. So this combination takes place. The process cannot go further, and the protein is not formed. When the protein is not formed, as you know, this protein is essential for the growth of nematode. This protein is not there, so this nematode will not grow and it will be killed or it will die. So the plant is safe. Okay, children, I think the time is over. I end the meeting.